I will discuss with you today Wallerian degeneration and the view that we have that it is reflecting the innate immune response of the peripheral nervous system to traumatic nerve injury or axonal injury. And I would like maybe to define first what is innate immunity. In general, we divide immunity into two. One is innate and the other one is acquired. The innate immune response is immediate, and characteristic features of the innate immune response are, for example, phagocytosis of tissue debris, pathogens, and apoptotic cells, and the production of cytokines. The acquired immunity is delayed response, which, and one of the characteristics, of course, is the production of antibodies. So here we are looking at tissue damage, and we are focusing on two features that are characteristic of the innate immune response, and that is the activation of phagocytosis by macrophages and Schwann cells, and the production of cytokines. So I would like to remind you, I have a question. Do you see the slide when the light is shining on the screen? Can you see the details here? Okay. So I would like to remind you that we discussed yesterday the result and the ability of CNS and PNS neurons to regenerate. And we heard from John and from what I told you that PNS axons readily regenerate, whereas central nervous system, adult central nervous system axons do not. And the experiment by David and Aguayo taught us that the ability to regenerate was not lost, but that the environment within the central nervous system prevented adult axons from regenerating. And in this experiment that I described to you in quite detail yesterday, it was possible to demonstrate that CNS axons can grow into a graft, a bridge made of a peripheral nerve, but once they get back to the CNS environment, they stop growing. So definitely something is different in the PNS from the CNS. So we turn to look again at the cellular composition of the peripheral nervous system, and I'll remind you, basically, what we have is axons which are surrounded by myelinating Schwann cells. In between fibers, we have fibroblasts. Once we cut the nerve at this region here, we can divide the distal segment into two portions. One is the injury site itself, where the impact occurred. That is where we have immediate tissue damage due to the trauma. And farther distal, we have Wallerian degeneration. And I told you that characteristic features of Wallerian degeneration are the breakdown of axons. Schwann cells detach themselves from the myelin portion of their membranes. Macrophages, which are scarce in peripheral nerve, normally are recruited from the circulation in large numbers, and as of the second day after the injury, they are activated along with Schwann cells to phagocytose and degrade myelin. And I ended up saying that this is a desired end result of Wallerian degeneration, because when we remove degenerated myelin, which is inhibitory to regeneration, we clear the path from this inhibitory element and we enable axons to regenerate back to their target cells. So we would like to further understand what and how Wallerian degeneration is regulated. And first of all, this is Waller. I would like you to know this person. And this is an original drawing of his in his publication on what we know now as Wallerian degeneration. Now, this is not a photomicroscope that was not taken by a camera. People at that time would look at a microscope and draw whatever they saw. So this is a drawing made by him of Wallerian degeneration. And since then, many studies have been performed, and we also added to the field of peripheral nerve injury and Wallerian degeneration. And the features that led us to suggest this schematic model are the following. First of all, we have here the intact nerve fiber, which is running here from the back of the screen towards you. We have the intact axons. You can see the filaments here. These are microtubules. We have here vesicular elements and mitochondria. Here is the Schwann cell and the myelin sheath, which is surrounding the axon. If we now perform a cross-section 5-6 days after the injury, we cannot identify anymore the axons, but instead we can identify one macrophage and two Schwann cells, and in the cytoplasm we have myelin debris as a consequence of being phagocytosed, and we can also identify lipid droplets, which is an indicator that these myelin particles are also degraded. So we look at the time course of myelin phagocytosis and degradation by making the following argument. If myelin is being degraded, then proteins which are specific to the structure of myelin, their content within the nerve should also be reduced. So we study the tissue levels of two myelin-specific proteins, myelin basic protein and P0, and you can see the time course of myelin degradation consequent to being phagocytosed by the macrophages and the Schwann cells. And already by 8 to 12 days, all myelin is gone. So we ask the question, how and why macrophages and Schwann cells phagocytose myelin in the first place? They must be activated. And to this end, we looked at, I'll jump over this, we looked at a particular molecule which is known as galactin-3, and previously, or when it was first identified and discovered, it was named MAF2. Now, I will not tell you in this lecture how galactin-3 activates phagocytosis. I will do it in a future lecture. I will only make the point that galactin-3 is activated in those cells that are actively involved in the phagocytosis of myelin, namely the macrophages that invade the peripheral nerve after injury, and Schwann cells that normally do not express myelin and the galactin-3 molecule, but following injury they do express it. And here we can see Schwann cells and macrophages isolated from such a peripheral nerve that undergoes Wallerian degeneration. And by immunocytochemistry, we demonstrate here, for example, the presence or the expression of galactin-3. So we made at the time the argument that if indeed galactin-3 is an indicator of activation of the phagocytic process, then we can follow the time course of galactin expression during Wallerian degeneration, and that will give us some idea about the kinetics of the activation of myelin removal through phagocytosis. So we start zero is when the injury is performed, and this is days after the injury. We start with undetectable levels of galactin-3 in the normal nerve. within 24 hours, we have already some upregulation due to Schwann cells that upregulate the expression of this molecule. But then, as macrophages are being recruited as of the second day, there is a very steep rise in tissue content of galactin-3. We reach a peak at about 5-6 days, and then gradually levels decline back to almost normal levels. So this is a transient effect, although macrophages and Schwann cells are still present. So this is downregulation that comes after the molecule is upregulated. Now, if we look at the kinetics of activation here and the kinetics of myelin removal, and we superimpose them, we can appreciate how well orchestrated these events are. And I would like to draw your attention to the fact that activation of the machinery that is responsible for removing myelin, the activation is preceding the onset of myelin degradation. Then we have myelin degradation, and that takes place when activation is at its highest levels. After myelin has been removed, activation is downregulated. So these events are very, very much orchestrated in time. We next turn to study another strain of mice that display a very unique feature. This strain of mice was discovered accidentally in an animal facility in England, where people, Perry and Brown, looked at Wallerian degeneration. They took these mice, they injured the nerve, and for some strange reason, the observation they made is the following. I will describe it to you here. They made a lesion here, and we have now a tissue damage at the site of the injury itself where the impact occurred. Now, when they look at the distal domain, they noticed that this distal domain does not undergo Wallerian degeneration as normal. It is extremely, extremely slow. So the term slow Wallerian degeneration is being used. And you can see here on top, these mice were first named Ola mice, but then WLDS, Wallerian degeneration slow. And in fact, when you look at this segment here, 1 week, 2 weeks after the injury, the axons look normal. You can take them out, put them on a stimulating electrode, a recording electrode, and you can record action potentials. It is true that it will not be exactly normal, but still, these axons are able to produce an action potential. Now, the evidence for this comes from the studies by Perry and Brown, and this is what we have also shown, that if you look at the injury site itself, there is evidence for tissue destruction. Here, macrophages are invading the site. They are actively involved in phagocytosis, but the domain that undergoes slow Wallerian degeneration, you can see that this was taken about 7-8 days after the injury. If I showed you this section and not told you that that was taken after injury, you would make the diagnosis that this is a normal intact nerve. But if you wait long enough, eventually, gradually, you'll get destruction as well. Now, if you look at the expression of the activation molecule, galactin-3, it is not expressed in this region here. It is expressed only at the site where you have tissue damage, invasion of macrophages, and activation of phagocytic activity. Here, the Schwann cells do not express, do not upregulate the expression of this activation marker. Now, if we take this distal nerve segment that undergoes in vivo slow Wallerian degeneration, in vitro it will undergo normal Wallerian degeneration. So it seems that there is something unique about staying in vivo at site to have this effect of delayed Wallerian degeneration, slow Wallerian degeneration. And again, when you have here Wallerian degeneration in vitro, these cells, the Schwann cells which are present here, will upregulate the expression of galactin-3. So again, we have a very good correlation between the expression of galactin-3 and whether or not phagocytic activity was activated in those cells. So here we have the difference between normal Wallerian degeneration and slow Wallerian degeneration. And here I put a list of events that are associated with normal and slow Wallerian degeneration. I could have chosen additional features, but I chose only 4. And if you compare those, you find out that macrophages are recruited normally, but recruitment is delayed in slow Wallerian degeneration. Galactin-3 is upregulated normally, not in slow Wallerian degeneration. Perry and Brown studied NGF. It is upregulated normally. It is not upregulated in slow Wallerian degeneration. And others studied neuropathic pain, which I'll define for you in a moment. It develops in what we would consider a normal rate in normal animals, but neuropathic pain will be delayed in its development in slow Wallerian degeneration. And what is neuropathic pain? We all know what acute pain is. This is a normal mechanism where we feel pain, sharp pain, if, for example, we cut a finger or we are being pricked by an injury. Neuropathic pain is a pathological condition where stimuli like touching the skin, which are normally perceived as even pleasant, will now be perceived as extremely painful. So this is just a simple example of what neuropathic pain is. Of course, it is much more complex from what I told you here. But here are the differences. So we ask the following question. What mechanisms could control normal and slow Wallerian degeneration to help us understand how these features are being regulated? So we now look at the possibility that Wallerian degeneration is in fact the innate immune response of the peripheral nervous system to traumatic nerve injury. And if that is indeed the case, then normal Wallerian degeneration results and is a manifestation of a normal innate immune response, whereas slow Wallerian degeneration results from and is a manifestation of a deficient innate immune response. So we set out to test this hypothesis, and we looked at 2 principal features. One is phagocytosis, that I will deal with in a future lecture, and the production of cytokines, which I will now tell you about in this lecture. So I'll start by presenting the end result and then the results themselves that led us to the following, what we define as the cytokine network of Wallerian degeneration. What I will show you is, or before that, sorry, what are cytokines? Well, cytokines are molecules that modulate the inflammatory response. We know about inflammatory cytokines They are responsible for the initiation and progression of the inflammatory response. There are anti-inflammatory cytokines that are responsible for down-regulating, shutting down the inflammatory response. Those cytokines are produced by innate immune cells, but also by non-immune cells. They interact with specific receptors that are present on the surface of immune cells, and thereby they regulate those innate immune functions that those cells will perform. But as you will also see, other cells, non-immune cells, will also respond to cytokines. So we studied 5 inflammatory cytokines: TNF-alpha, IL-1-alpha, IL-1 beta, IL-6, and GM-CSF, and 1 anti-inflammatory cytokine, interleukin-10. And the end result of this was the cytokine network of Wallerian degeneration. We'll run through this, where axotomy induces first the production of TNF-alpha and interleukin-1-alpha. These cytokines will induce the production of interleukin-6 and GM-CSF. Then interleukin-1 beta is produced, and it contributes also to the production of IL-6 and GM-CSF. Then IL-10 is being produced, and IL-10 downregulates the production of all cytokines and the production of itself as well. And that is how the entire event comes into conclusion. So it is extremely important that an inflammatory response will be turned on, but also be turned off. We cannot have tissue repair without inflammation. But if inflammation is not controlled, it becomes a disease by itself. So we need very rapid upregulation of the inflammatory response. It should be controlled and then turned off at the right time. And if we look at these cytokines in context of cells that produce them, the following can be observed. Here we have the axon in green, surrounded by a single Schwann cell and a fibroblast. These are the cellular elements in the intact nerve. If you cut the axon, Schwann cells respond first by upregulating TNF-alpha and interleukin-1-alpha. They make fibroblasts produce interleukin-6 and GM-CSF. Then interleukin-1 beta is upregulated again in Schwann cells, and macrophages are recruited. These macrophages are induced to produce the inflammatory cytokines, but at reduced levels. They will produce interleukin-6 and then interleukin-10, and interleukin-10, as I told you, will then turn off, down-regulate the production of all cytokines, including the production of IL-10, and the entire inflammatory event is turned off. Now, to quote Jack, the question is, how do we know? So that is how we know. We start with testing the production of cytokines, and we use 3 different methods. One is to look at transcription, which is to look at messenger RNA levels. Second is to look at translation, which is to look whether or not the cytokine protein is synthesized. Third is to find out whether cytokines are in fact secreted into the extracellular space, since they have to diffuse and act upon neighboring cells in a paracrine and sometimes autocrine fashion. And finally, we would like to know which cell type produces each of those cytokines. So I'll start with the inflammatory cytokines, TNF-alpha, interleukin-1-alpha, and interleukin-1 beta. And you can see that TNF-alpha, the expression is already there in the intact preparation, and it is upregulated following injury. Interleukin-1-alpha has some levels of expression. Normally, expression is upregulated after injury. Interleukin-1 beta, the message is not detected normally, but it is upregulated after injury. We now look at the protein at the tissue itself by immunocytochemistry. First, TNF-alpha. Some expression can be identified already in the intact preparation, but there is a very rapid upregulation of this expression after injury, 5 hours, 12, and so on. Interleukin-1 alpha is not detected, the protein is not detected, but upregulated very rapidly. Interleukin-1 beta, the protein is not detected normally, not 5 hours and not 12 hours after the injury, but as of 24 hours and later on it is detected. I will superimpose now on this the expression of the message. So you can see here a very tight correlation between message expression, which is plus, and the immunocytochemistry telling us that the protein is there. And that is for TNF-alpha. If we look at interleukin-1 alpha, there is a discrepancy in the intact preparation. We have the message, we don't have the protein, no translation. If we look at interleukin-1 beta, we have no expression, no message in intact, but the message is upregulated already 5 and 12 hours after the injury, but the protein is not produced. It is produced anywhere between 12 to 24 hours. So we have a discrepancy between message and translation of the protein itself. What it tells you is that if we had only looked at the message, we would have gotten a completely different view on the production of cytokines. Because what really matters is the protein and not the message. Because proteins do the job, not the message itself. So it is extremely important in the case of cytokines and probably other molecules as well to look both at message and translation and not assume that if you have the message, it means that you do also have the protein at all times. So we now look and try to identify which cells produce each of those cytokines. So we isolate those cells from the Wallerian degenerative nerves. Here we have Schwann cells, macrophages, and fibroblasts. We know that these are these cells because of their morphology, but also because they have unique antigenic characteristics so that we can use immunocytochemistry to help determine whether you are dealing with a Schwann cell, a macrophage, or a fibroblast. I will not go into the details. It is all written here in the table. But as you can see, all cytokines are being produced by Schwann cells. All cytokines are produced by macrophages. Fibroblasts may be producing a little bit of TNF-alpha, not IL-1 alpha, neither IL-1 beta. And if you look at the message of those isolated cells, you can find out that all of them will have the message will have the message, Schwann cells and macrophages will have the message of those cytokines as well. So let us now turn to slow Wallerian degeneration. And I would like to draw your attention that slow Wallerian degeneration is like an experiment that nature designed for us. It made an animal which can be injured, but it is not responsive as the normal mouse. So we look at the production of cytokines in slow Wallerian degeneration, and we look at what one can find at the injury domain itself and the more distal portion that undergoes slow Wallerian degeneration. So the injury domain is up here, and you can see that at the injury domain, where we have tissue damage, where we have macrophages, where we have phagocytic activity, we can also detect TNF-alpha, IL-1 alpha, and IL-1 beta proteins here in that site. However, if we look more distally, where slow Wallerian degeneration takes place, we cannot detect any of those cytokines. We were first puzzled, so we repeated those experiments and made certain that we always take the same segment and compare the injury site and the slow Wallerian degeneration domain from the same animal side by side, and we repeated it time and again, and always the results are the same. Now, if you look at message, you find that in the slow Wallerian degeneration domain, the message is upregulated. So if we now superimpose message expression on top of protein expression, you find that there is a good correlation at the injury site, but discrepancy here where slow Wallerian degeneration occurs, making again the point that had we only looked at message, we would have gotten, or we would predict a completely different interpretation than from what really takes place here in the liver. There is no upregulation of protein production of those cytokines in the slow Wallerian degeneration domain. So we move now to GM-CSF, which is yet another inflammatory cytokine. It is known as GM-CSF because it is also involved in the hematopoietic system in the differentiation of macrophages and granulocytes, as the name indicates. So here we have evidence that the message is not expressed normally but upregulated after injury. If we look now at the time course of GM-CSF production after injury, the red here is normal Wallerian degeneration, and you can see that production is very rapidly upregulated within 5 hours, and this is the time course. Here in blue, production of GM-CSF in those slow Wallerian degeneration domains. There is some production, but levels of productions are significantly and substantially lower than from what we find in a normal mouse. And if you look at cells and ask the question, which cells are responsible for the production of GM-CSF, we isolate cells from these preparations. We find that macrophages contribute very little, if any. Schwann cells contribute also very little, if any. But fibroblasts produce lots of GM-CSF. And that makes sense, since here we have a very rapid onset of production within 5 hours. So it could only be a cell which is normally residing in the tissue that is producing GM-CSF. And fibroblasts are there at all times. So you have the cell responsible for the production, the rapid production of GM-CSF. Next, we move to IL-6. IL-6 is not expressed normally, but expression is upregulated after injury. We next look at the time course of IL-6 production, again in red, the normal mouse, and now production is being biphasic. Extremely rapid onset, in fact within 2 hours, we already detect IL-6 protein, one peak, then the second peak, and downregulation. Now, here we have in slow Wallerian degeneration, again, some IL-6 is being produced, but substantially lower levels. We then ask the question, which are the cells that produce IL-6? We find that macrophages produce IL-6, fibroblasts produce IL-6, Schwann cells no. Now, with this knowledge, we turn back to this figure here. Fibroblasts are responsible for the production of the first phase, since they are present there at all times and they can respond very quickly. The second phase correlates very well with the kinetics of the recruitment of macrophages to the nerve. So the second phase is due to the induction and production of IL-6 by the invading or recruited macrophages. We now turn to IL-10. IL-10 is not produced normally. It is not produced early in Wallerian, the expression of the message is not upregulated in the early stages of Wallerian degeneration, maybe little bits here, but definitely at later stages of Wallerian degeneration. We look at protein production. In red, we have again normal Wallerian degeneration. We detect production early on, but extremely low levels. Levels of production increase as of the third day after the injury and reach a peak at 7, and then levels are downregulated. Here we have some production, but low levels of production throughout in slow Wallerian degeneration. We again ask the question, who's producing IL-10? And the answer is that macrophages produce IL-10, fibroblasts produce very little, Schwann cells do not produce at all. We go back now to this figure here. These initial low levels of production are attributed to fibroblasts, which are present there at all times, and are producers, but poor producers of IL-10. Highest levels of IL-10 production are produced by macrophages that are recruited to the tissue with a similar time course. So these are responsible for the later phase, or the second phase of production of IL-10, the high levels of production of IL-10, which now brings us back to the cytokine network that I suggested to you in the first place. What we have seen is that axotomy first induces the production of TNF-alpha and interleukin-1 alpha. Then we had IL-6 and GM-CSF. Then we had IL-1 beta that contributed to the production of IL-6 and GM-CSF. Finally, we had the production of IL-10 that downregulates the production of all cytokines. And when we turn back to asking the question about production in context of the cells that are responsible, that do the job of producing cytokines, we find out that Schwann cells are the first to respond to the injury. And when you come to think about it, it really makes sense. Who other cell is in such close and intimate contact with the axon to respond immediately to an event of injury that the axon undergoes? You would assume that that should be the Schwann. And indeed, the Schwann cells are the first to respond. In fact, TNF-alpha message and IL-1 alpha message are already sitting there at all times. They are ready. It's like having the engine on, but you didn't press yet the gas, and you did not release yet the handbrake. But once you release it, the engine is already running, and you can start racing. Then these cytokines will induce the very neighboring cells. Who is the next neighbor? These are the fibroblasts normally. So they are induced to produce IL-6 and GM-CSF. Now, for some reason which we do not understand, there is a lag in the production of IL-1 beta, but then these cytokines, with the help of chemokines, the point that I will make in just a moment, help recruit macrophages. These are induced to produce the cytokines, the inflammatory cytokines, IL-6 and IL-10. And as I told you, IL-10 finally downregulates the production of all cytokines, so the inflammatory response is turned down. Now, if we look at slow Wallerian degeneration, what do we miss in slow Wallerian degeneration? We miss TNF-alpha and IL-1 alpha. I'll show it to you again. We miss the upregulation of TNF-alpha and IL-1 alpha, which were responsible for initiating the whole cascade of events. So if you don't have TNF-alpha and IL-1 alpha, you have nothing of those. So you remain with what looks like an intact axon, an intact Schwann cell, and a normal fibroblast. And that is exactly what we identified when we cut cross-sections through a slow Wallerian degenerating nerve at 1 week and 2 weeks after the injury. So now we turn back to the list that I made, and I would like to try and answer how those cytokines could regulate and be responsible for the differences that are seen here on the board. So we start with macrophage recruitment and galectin expression upregulation. So we know from the literature that TNF-alpha, IL-1 alpha, and IL-1 beta themselves act as chemoattractants to macrophages. We also know from the literature that these cytokines will upregulate the production of a chemoattractant MCP1, and combined they will help recruit macrophages into the nerve. So in the presence of those, you have recruitment, and in slow Wallerian degeneration, in the absence, you do not. Next, we have shown that GM-CSF, one of the cytokines that I discussed with you, is responsible for upregulating the expression of the activation molecule galectin-3 in macrophages and Schwann cells. So when you have GM-CSF, which is the case normally, you have expression of galectin-3, and in slow Wallerian degeneration, where you have extremely low levels of GM-CSF, galectin-3 is not upregulated. We now look at this in the context of the cellular elements that we find, then we can say the following. Here we have, after axotomy, the production of the inflammatory cytokines. They make fibroblasts produce GM-CSF. As a result, the activation molecule galectin-3 and phagocytosis are upregulated in Schwann cells. Then additional cytokines and the chemokine MCP1 is upregulated in fibroblasts. Then macrophages are recruited. They are activated to upregulate galectin-3, phagocytosis in IL-10, that then turns down the entire array of events. Now, how about NGF? I told you that NGF is being produced normally, but not in slow Wallerian degeneration, or extremely low levels are produced here. So again, after injury, we have the production of those inflammatory cytokines. Apparently, those cytokines will induce fibroblasts to produce NGF. Fibroblasts produce NGF during Wallerian degeneration. And NGF is also produced by Schwann cells. The exact mechanism responsible for upregulating the production of NGF in Schwann cells is not known. But what we do know from yesterday is that NGF will help keep cells alive, and it will also promote the cells that are responsive to NGF. And in the peripheral nerve, we have such cells. We have the subset of dorsal ganglion cells which are responsive to NGF, and we have also sympathetic neurons which are responsive to NGF. So this is another feature of Wallerian degeneration that helps to promote regeneration on top of clearing inhibitory myelin. And finally, neuropathic pain. We know from the literature that TNF-alpha and IL-1-alpha and so also IL-1-beta will make sensory neuron fire spontaneously. Now, if they fire spontaneously, messages are being transmitted to higher centers, and it is perceived as pain, extreme pain. Now, we know also that these cytokines will induce the production of NGF, and it was shown that NGF can sensitize the nerve endings and receptors that respond to various stimuli like mechanical stimuli and pain stimuli. So, and finally, it was also found that IL-6 deficient mice display reduced neuropathic pain. So what you would expect then is that in normal mice, where you have the production of all of those cytokines, that neuropathic pain will develop, whereas in slow Wallerian degeneration, where you have reduced levels of production or no production at all, that neuropathic pain will develop with a delay. And this is how it can be visualized schematically. Usually in neuropathic pain, one of the ways or methods that people who study neuropathic pain, induced neuropathic pain, is to produce a partial lesion. What does it mean? We have here two axons. Only one is injured and the other one is not. Then you have the production of those inflammatory cytokines in the injured nerve. Fibroblasts are induced to produce NGF, Schwann cells as well, and all of those NGF will now sensitize receptors associated with the intact fiber, which is situated nearby anatomically. Next, these cytokines will induce spontaneous firing in the intact nerve, which is adjacent to the one that was injured. The result is neuropathic pain. So in the absence of all of those, neuropathic pain development will be delayed. So you can see that cytokines can help understand how various events that are associated with Wallerian degeneration are controlled. And that is what I just said. So I hope that I was able to provide you evidence that Wallerian degeneration is indeed the innate immune response of the PNS2 injury, mostly since innate immune cells are involved in the process, macrophages. Innate immune molecules are involved, cytokines and chemokines. Innate immune functions are involved. Phagocytosis, for example, which I will discuss with you in a later lecture. And therefore, normal Wallerian degeneration is indeed a manifestation of a normal innate immune response, whereas slow Wallerian degeneration is a manifestation of a deficient innate immune response. Thank you.
Oligodendrocytes and Schwann cells, apart from the I want, what is the difference between Schwann cells and oligodendrocytes?
Well, all what I can tell you about oligodendrocytes compared to Schwann cells is the following. And I'm afraid that there is very little comparison that can be made because oligodendrocytes were not studied in such detail. What we know is that after injury in spinal cord, for example, those inflammatory cytokines, which we believe are responsible for setting on the entire cascade, these inflammatory cytokines are not upregulated after injury in the spinal cord. So here we have some resemblance to slow Wallerian degeneration, if you want. However, whether or not and how oligodendrocytes produce cytokines, this was not looked at such detail, so I can't really tell you about that. What I can also tell you is that oligodendrocytes are not involved in taking up degenerated myelin, whereas Schwann cells do. Oligodendrocytes in correlation do not upregulate the production of galactin-3, which I will show you, an activation molecule for phagocytosis, whereas Schwann cells do. So there is a difference between oligos and Schwann cells in the way that they do respond to injury, but we do not have the entire picture available.
About interleukin 10 or the graph, you said the interleukin, the small amount is produced by the fibroblast, then majority produced by the macrophages. But on the graph it says the interleukin 10, it starts at a higher point, then it gradually slows down and then it tends to go up. Is there any down regulation?
You're asking about this here?
Yeah.
So, I mean, the initial levels that are being produced are not different between normal and slow Wallerian degeneration. Okay? But as you can see here, these are extremely low levels of IL-10. These low levels do not interfere with the production or the generation of the entire cascade of events. There are probably levels are not sufficient to down-regulate, hold down the activation or the production of the inflammatory cytokines that I showed you before. But here, when you reach extremely higher levels of IL-10, then they become effective and they down-regulate production.
Yes, sir, but the question is now initial segment is governed by the
Fibroblast.
Fibroblast. So it starts at a higher point, then it slows down according to the graph. So is it due to the...
Oh, here,
No, there are the start point. Yeah.
if it starts at lower levels,
Yeah, the initial point itself. Now you
well, this is not initial point. It is the first time we can ever detect it.
yeah, then it gradually goes down and then it slows.
Well, this is not significant. This is not significant. I mean, these points are not statistically significant one from the other. But it looks like it goes down, but it's not.
Yeah, so I was thinking whether they are saying down regulation of that activation of the fibroblast and the result it goes down.
No, we cannot tell, but because statistically we cannot tell that these 3 points differ one from the other. But certainly this are obviously. Yes.
I was wondering that the treatment for peripheral neuropathy, for diabetic neuropathy, several drugs are being used. Like we have gabamylotics, like gabapentin, betaine, etc., including duloxetine, which is not related to that group. So I was wondering how they which mechanisms are affected when they relieve the neuropathic pain. That is one question. And recently they have said that the electromagnetic frequency stimulation does help in relieving diabetic retinopathy. So wanted to know if you can associate the mechanisms that are producing pain and the mechanisms which would help in relieving the pain.
Well, as I very briefly alluded at, neuropathic pain, or I will start in a different way. Neuropathic pain is the final outcome of many mechanisms that can end up having the same clinical manifestation. It can be peripheral, it can be at the level of the dorsal ganglion, the spinal cord, and even higher levels. So definitely one side is the peripheral nerve, there's no question about it, but it is not the only side. Second, definitely inflammation is a very important factor. So there is even in the literature a term, inflammatory neuropathic pain. So inflammation can be caused, or chronic inflammation can be caused by many, in the pathophysiology of many diseases. So when you have chronic prolonged inflammation, many times you get also neuropathic pain. Now we don't get it always because different people respond differently because of the genetic makeup, which is also a factor. Now, with respect to diabetic neuropathy, it can be involved also. It can also involve, of course, neuropathic pain. Now, I do know, although I'm not working in the field, and so I'm not an expert. So please take that into account when I'm responding. Anti-inflammatory treatment, of course, are tried on such patients using antibodies against inflammatory cytokines. I know that IL-10 was used in patients. I don't know how well it was received, and so on and so forth. But again, I'm not an expert in the field of neuropathic pain, and I hesitate to make any other statement. With regard to electromagnetic fields, I'm completely ignorant, so I won't say a word about it.